Welcome everybody, and thank you for asking me to give this talk. I'm a scientist, so I've been working with Aspergillus and the individuals of Aspergillus for about 25 years now. I came to Manchester in 2005 to work with David Denning, and what was just the beginning of the National Aspergillosis Centre back then. It hadn't been formed at that time. So Beth asked me to talk about the genetics underneath Aspergillus and aspergillosis. Why do people get it, and what's going on, and what do we know about it at the moment? So I'm Professor of Infection Genomics in the University of Manchester, and I look at the genomes of people who catch diseases, and also the genomes of the pathogens themselves. And about 13, 14 years ago, we started to look at the genomes of people who had come into the hospital with various types of aspergillosis. And we wanted to compare them and see what the differences were, and see if we could find some factor that underpinned why they got aspergillosis and other people didn't. So I'm going to start off quickly by just sort of laying the table here and showing you the background. And you're all familiar with this kind of figure, and this is obviously some kind of AI-generated image of a lung, but the basic...
Oh, the slide hasn't changed? We're still on the title.
Oh dear. See, it's making... Okay, see that?
Yeah.
Okay, all right, I'm gonna have to do this every time, I think. Okay. So the airways in everyone are very similar. You've got a big tube coming in, and it branches and divides, and the tubes get smaller and smaller and smaller. What this means is that the particles that are breathed into the lung get deposited at different levels. And the cells that are in the airways of your lung are different at every level. So the cells that are in your nose and mouth and the top of your throat here, it's quite complicated. There's lots of different types of them. If you look on the right-hand side of this very complicated diagram, if you look on the right-hand side, you can see on that side there, representing the cells that are in your nose and in your upper airways. They're all quite different and very complicated. As you get a bit further down, where the lungs start branching and the airways get thinner and thinner, the cells get simpler and simpler and smaller and more delicate. So it really matters to the progress of the disease and the type of disease you get, how far down the spores can get. Okay. I'll just run it through. So this is, again, it's a very complicated picture, and ignore the graphs for the moment. The graphs are there for the people who want to know the numbers. The story is what's important here. So again, if we look at our simplified diagram of the airway, starting on the left here, you see that lots of these big complicated cells, and they get smaller and thinner and more delicate as you get right down into the bottom of your lung, and these very tiny passages and the alveoli, these little sacs that sit at the end of them. On top, the first thing is on top of the cells in your lung and all through your lungs, there's a layer of mucilage, and this is what sputum is made of. The mucilage is waved. Every cell has these little cilia that wave and beat thousands of times, and they push the mucilage up through the lungs, and it comes out. You either swallow it or it comes out as sputum, and you cough it up. This is incredibly effective. Mucilage captures spores very well because it's very sticky. So almost everything you breathe in is just hoovered up and pushed up your lungs until it comes out. And this is a very effective way of getting rid of pathogens. Everybody has it. It does mean that each one of us has fungus in our lungs all of the time, because we're continually breathing in spores, and the spores are germinating. You can see there's a couple of spores germinating here on this slide, the green ones. in the mucilage. So we've all got these tiny little colonies of fungus in our lungs. It's perfectly normal. Every single person has it. Sometimes those spores will actually get through that mucilage layer. And you can see again on the left of the diagram, you'll see that some spores are effectively eaten by the cell layer that they meet. They're very, very good. Your human lung cells are very good at ingesting and digesting fungal spores and other types of bacteria and so on and so forth. And usually that protects you completely from fungal infection. However, that doesn't always work. Even in healthy people, that doesn't always work. And sometimes the spores escape and they'll germinate and you get these long thread-like growths going through the cell layers, and they can sometimes penetrate and get deeper and deeper into the tissue, and you get these kind of slight, small, tiny colonies of fungus. And again, perfectly normal. Probably every single person in the world has about 5 to 20 of these tiny microcolonies in their lungs at any given time. So perfectly normal to have fungi in your lung. It's only when this gets a little bit out of hand does it become a problem. So what we've been looking at is what goes wrong in the lungs to allow the fungi to germinate and penetrate and grow more effectively. What has to fail in this situation for the fungus to succeed and for the defenses in the airway, these really effective defenses? What has to happen for them to fail? And again, you can look at the graphs if you want. The numbers are all there, but don't worry about that for the moment. So we started by here we go. We started by looking at the genomes of people who are coming into the National Aspergillosis Centre, and even before then. And you can see the numbers of patients on the right. So we started with 156 chronic pulmonary aspergillosis genomes, 232 genomes from patients with ABPA, and we had a couple of hundred asthmatics as well as controls, and there's a lot of healthy people being genome sequenced now as well. So this is round about back in 2012. This is a lot of people for an aspergillosis study. It's not really very big for a genetic study, but in terms of the number of patients who come through the Aspergillosis Centre, this is quite a lot of people. So we sequence the genomes of all of the people who come through the Aspergillosis Centre, and we compare them to our control groups, healthy people, people with asthma, and so on, comparable conditions. This is a lot of data, and it's very difficult to interpret. But if you look at the people on this call, on the Teams call, you can see we're all pretty similar, aren't we? We've all, we've got 2 eyes and 2 ears and a nose, and we all, generally speaking, people are pretty similar. So we've got 3 billion DNA bases in our genome. That's what codes for a human. Most of it does something. Don't think about these ideas of junk DNA and so forth, but most of the DNA that you have will do something. And short stretches of that DNA code for proteins, and we call these genes. So you have about 25,000 genes in your genome. Each one codes for a protein, and the protein just goes off and does something in the cell. That's all you need to know about that. So the idea here is we're going to compare our population with people who've got aspergillosis against another population, and we're going to see if we can spot some differences. And again, if you look at the people who are on this call, and again, okay, 2 eyes, 2 ears, a nose and a mouth, but actually you can tell the difference between everybody, can't you? So we've got 3 billion base pairs of DNA. Probably each person has 1 million differences when you compare it with the next person. So this is quite a big computational task. We've been doing this for quite a long time now. Sorry about this. Here we go. And we have been discovering variants in individuals with disease. Sometimes it's not the way we expect. And again, ignore the graphs, don't worry about those. We found the first one about 7 or 8 years ago, and it's in a gene called ZNF77. You don't need to worry about what that does. It's a control gene. And anyone who has this variant in that gene has about a 16-fold higher fungal burden in their lungs. And that's really, really very significant when you're thinking about fungal disease. We found that 60% of people with ABPA who have the high Aspergillus burdens in their lung. So sometimes with ABPA, you don't have much Aspergillus in your lung, but quite a number of people with ABPA have really quite a lot of Aspergillus when you look at the burden of Aspergillus in their lungs. So 60% of those individuals carried this variant. It can be present in healthy people as well, though, and this confused us for some years. Asthmatics carried this variant, and healthy individuals carry this variant as well. And it's present in about 4% of the normal population, the healthy population. Those people have got lots of fungus in their lungs, but they are not ill. So obviously with ABPA, and I'll talk a little bit more about this later, you've got to have asthma, you've got to be allergic to the fungus. And if you don't have those things, you can still have lots of fungus, you just won't have ABPA. So whilst this is a really important gene and genetic cause of ABPA, it's not the whole story. You just have to think about the context a little bit. What does that do? What's the consequence of having that variant? The first thing is, I'll just show you this quickly. Again, I don't like to show you lots of graphs, but the Aspergillus in your lungs actually grows faster. Was the first thing we noticed. It germinates quicker, it grows faster. That all fits in with having more Aspergillus in your lung. When we looked a bit more closely, we were able to see that in normal people, and this purple plane on your screen here is your epithelial layer. It's an epithelial layer from a normal, healthy type of individual. You can see all the cells. It's quite hard to see the individual cells, but you can see they're all very well stuck together. It's a continuous layer. It's completely watertight. It's really tough. And if you remember from before, each one of those cells is very good at eating fungal spores. So it's a really effective way to stop infection. When we looked at this variant we found, this ZNF77 variant, it's the next panel there on the middle of the screen. You can see there's lots of holes in it. And it's on the graph, again, to the right there, you can see it's incredibly leaky. The blue line shows the leakiness. So this is a really badly formed epithelial barrier layer. And this is very likely to allow the fungus to grow into the tissues underneath the epithelium very easily. So not only does it help the fungus grow, because these cells are breaking and releasing lots of nutrients that the fungus likes, so the fungus grows better, but it also changes this barrier and stops it really being an effective barrier at all. Okay, so that's the first variant I wanted to talk to you about, and that's an ABPA variant, and I'll talk about the numbers a little bit more in the next few slides. So let's go back again to our airway colonization, how fungi get in and how they're cleared away. And you can see that the very first step, right on the left of this diagram, those big cells, they're very able to eat the fungal spores. They just phagocytose them, that's a technical term for it. They completely enclose them, they drag them in to these vesicles, and then they digest them using acid. It's really very dramatic. I'll show you a picture in a minute. And it's a very effective process. Sometimes that goes wrong. So the next way that the fungus can get around this system is by evading that process, or by this digestion process not quite working correctly. So these are actual pictures of a fungal spore. That's this red round thing there on the top left. being eaten and digested by a human epithelial cell. And as you can see over time, we've put some dyes in here. There's little green spots here. They're stained with something which picks up something called a lysosome. There's a little pockets of digestive material in your cells, and they get attracted to this what's called a phagosome. That's where the fungal spores being taken up. And you can see that the edges of this phagosome get greener and greener, and there are less and less of these little green dots as they are attracted to the phagosome, and they're releasing their digestive contents into the phagosome over time. It's quite quick. It takes only an hour, this whole series of photographs, until there is none of these little lysosomal particles left, and the fungus is dead. The spore is completely dead. Sometimes this doesn't work. Again, it's a little difficult to see on this bottom panel, but sometimes the fungus can germinate and escape from this process, and that leads to infection. So the second gene we were looking at was a gene called EEA1. Now, this is responsible for this acidification of these phagosomes, this digestion of the fungal spore. And we discovered that about 40% of the individuals with ABPA have some kind of variant in this gene. It's not the same variant. There are several different variants scattered across the gene. So it's a slightly more complicated situation, but it's actually quite common in ABPA. So this was the second one that we found. And again, this is an ABPA variant, really. Specifically for ABPA, and this does not occur in healthy individuals. So that's the second one that we found. So it fails, people who carry this variant aren't quite as good at digesting those fungal spores. Okay. The final one I want to talk about is, it's new. So we've not talked about this before, ever, to anyone. And this is to do with chronic pulmonary aspergillosis. And we've discovered that variants in the ITGB3 gene, I'll talk about what that is in a minute, are associated with CPA. So that if you carry this variant, about 35% of chronic pulmonary aspergillosis patients have this variant, which is quite significant for this type of thing. So that's a major gene that underpins susceptibility to chronic pulmonary aspergillosis. It virtually never occurs in healthy individuals, so this is a really strong difference. And again, this is a tissue integrity or a repair phenotype, same as we saw with those epithelial cells with holes in. This gene is involved in how cells stick together, and it's or how they repair and fix themselves. So it's a really important physical attribute of the lungs of people with chronic pulmonary aspergillosis. This is it in more excruciating detail, I'm afraid to say. There we go. The integrin ITGB3 is this green protein here, sat in a membrane. It leads, when it's not controlled, it leads to fibrosis because it over repairs. It keeps trying to repair tissue damage. It keeps trying to make junctions between cells. So it leads to a very sort of scar-like phenotype in the lung, and it causes a lot of fibrosis. And you'll hear words like bronchiectasis and bronchitis and things like this, which are the result of this buildup of scar tissue and harder tissue in the lung, which they're really important components of chronic pulmonary aspergillosis, also ABPA and other types of aspergillosis as well. So if this protein is not controlled well, you get fibrosis. We know that one of the things that happens in the cell, and one of the things that this controls, is this CYR61 protein. Now this, again, is involved in our disease profile. And this is the most significant protein in chronic pulmonary aspergillosis patients. Normally, And if you look at the bottom left here, and again, I'm going to ask you to just think about this graph a little bit. When you challenge the cells of a healthy individual with Aspergillus fumigatus, and you just grow them in a dish and add some fungus to them and watch what happens. This is the protein that it's increases in amount 10,000-fold. It's the highest increase in any protein in the lung during infection. However, if you look at the left of that slide where it says CPA, people with CPA do not respond. And we think, we've not directly linked the dots yet, but we think it's because the ITGB3 is not signaling properly. There are many, many other changes. On the right, you can see all of the immune genes, and green means that they're turned up and red means they're turned down. Lots and lots of changes, but none of these have genetic differences in them. The levels of them change dramatically, but it's not actually them. They are being controlled from somewhere else. And we think that the master control is coming from this ITGB3 and CYR61 pairing. And they're proteins that are involved in tissue repair, scarring, and regeneration of tissue. So every time you cut your finger, these proteins will help you heal. Right. Just to summarize then, so that was a lot of quite complicated information, I realize, and I apologize for that. It is quite a complicated thing. We've got quite a lot of information there. We know quite a lot, and these are quite important genes. You can see that we're hitting some really quite big numbers in terms of the amount of disease they explain. And for genetic causes of disease and for genes and so on, these are big numbers. So normally when we look at variants that cause disease, you don't normally get these numbers. So these are important. But you'll see there's lots of question marks on this slide. So if you look at invasive pulmonary aspergillus here on the top, we don't know whether any of these variants are involved in that disease. We've just not had the chance to look yet. Similarly with ABPA, we don't know about ITGB3. CPA, we do not know about ZNF77 or EEA1 yet. Bronchiectasis, we probably can see the ITGB3 in it, but we don't know anything about ZNF77 or EEA1. And we do know quite a lot about healthy individuals, but sometimes you can get these variants in healthy individuals as well. So this is a complicated story, and there's lots of blank spots. But we are getting to an interesting situation where maybe we want to think about starting to screen, or thinking about what we should do with this information. This is actually the brief that Beth gave me for this talk, talking about dodgy genes and should I be worried? Yes is the answer to dodgy genes. Should I be worried? I'll talk about. I think again it's extremely important to say I'm a scientist. I'm not a clinician or a genetic counselor. So the should I be worried is it's kind of not my job. I'm going to give you some numbers and say what I think about it, but that's only my opinion. So just please be aware of that. So to summarize, what have we got? Where are we now? And the first thing to be aware of, if you think right back to that original slide where you've got all that fungus coming into the lung and getting cleared by the mucilage or sticking and getting in, it's got to be that fungal disease. If you end up with fungal disease, it's a series of unfortunate events. Most of them are not genetic. So that's the very first thing to say. So you've got to be a bit unlucky. So if you've got IPA, invasive pulmonary aspergillosis, if you look on the left, some kind of immune suppression is necessary, and probably about 5 or 10% of the population have got that. It really needs to be a bit longer term than, short term is not necessarily a risk. A long term, so 20%. So maybe the at-risk population for this is possibly 1% of people in this country. With chronic pulmonary aspergillosis, you almost definitely need an underlying condition like tuberculosis or COPD. And the incidence of those is quite low. COPD is, one of the highest, and that's 2.5% of the individuals in this country. And you probably need to have actual lung scarring and damage arising from these conditions, which occurs in maybe about 30% of the cases. So again, your at-risk group is about 0.7%. And then with ABPA, you've got to have asthma and you've got to have fungal sensitization. So asthma is 6.5% of the population. 4% of the population are sensitized to fungi. So again, your at-risk group for ABPA is 0.25%. So when you think about a question like, are my kids at risk when I pass this down, you don't often see familial invasive aspergillosis, chronic aspergillosis, or ABPA. It does happen sometimes, but it's very rare. And it's probably for this reason, because the risk factors, the non-genetic risk factors just don't happen to your kids or to other people. You've got to have this series of events that happened that aren't genetic at all. And then, after that, yes, the genes will become important. So my opinion is, well, they're very important. If you're in one of these at-risk groups, I would suggest, yes, the genes are very important. If you're not, and if you have not gone through any of these risk events, then probably you don't need to be too worried if you carry one of these gene variants. But again, those are considerations that will be worked out by the clinicians and people who do genomics testing, because there's complex calculus of whether it's worth doing the test, how many people will be affected or helped, and I can't speak at the moment. Okay, so I'm going to stop there. I'm happy to take questions. I've got to say that this work has been supported by a huge variety of people. Obviously, the National Aspergillosis Centre, non-cash and other type of contributions from many clinicians in the NHS and the NHS themselves. It's been funded by the European Union. It's been funded by the Medical Research Council. I work in the Manchester Fungal Infection Group, who are a marvellous group of scientists based in the university, and they're tremendously supportive and helpful. But I really particularly want to mention the Northwest Lung Centre charity at Wythenshawe and the Fungal Infection Trust, who are pivotal in funding this type of work, because sometimes you need a small amount of work doing, and you've got a gap in your research, and these two funding bodies are tremendously helpful for getting you through those times.